Welcome and thanks to everyone joining us today for the inaugural Imugene Science Series, where we want to give our stakeholders the opportunity to go a bit deeper and learn more about the people, history and details behind Imugene science. Today we're going to focus on Imugene's oncolytic viraltherapy, known as CHECKvacc, which recently saw the first patient dosed as part of a first-in-human phase 1 single-centre dose escalation trial for patients with triple-negative breast cancer. Principal investigator for that trial is Dr. Yuan Yuan, Associate Professor specialising in breast oncology in the Department of Medical Oncology and Therapeutics at City of Hope. Joining her today is the inventor of the technology, Professor Yuman Fong, the San Giacomo Family Chair in Surgical Oncology at City of Hope. In terms of some quick housekeeping, the initial presentations will last for approximately 20 minutes before we would welcome any questions. If you do have a question, please enter it using the Q&A panel within Zoom. To kick off proceedings, I'll first hand it over to Imugene's CEO and Managing Director, Leslie Chong.
Hi, everyone. Welcome for joining us. Thank you for joining us today. I just want to remember or note that today is Remembrance Day. Tomorrow in the States will be Veterans Day, and the day after it will be my 30th birthday. So I'll move on with a little joke. We have 3 platforms here, and the one that we're talking about today, where we already have a patient on, is CHECKvacc. This is our oncolytic virus with a PD-L1 transgene, and I'll just go quickly to Dr. Fong.
Thank you very much, Leslie, and thank you for all of you for joining us and engaging in this dialogue. I just want to first talk about how oncolytic viruses work. The whole idea of oncolytic viruses is that we are relying on a virus that likes and almost exclusively only infects cancer, and after infecting the cancer, replicates within the cancer, and then lyses the cell, so explodes the cell so that the viruses that are created within could go and infect other cancer cells nearby. And the whole idea of this is that many viruses only like infecting certain tissues. So hepatitis viruses only infect liver, meningitis virus only infects nerves. And what this field does is trying to design viruses or find viruses that only infect cancers. We are going to rely on these viruses to kill cancers directly, but we're also going to rely on these viruses to turn on the local immune environment of the tumor to allow the host's, the patient's own immune system to recognize cancer better and to attack it. And the hallmark of all of this in terms of turning the local immune environment much more immunogenic is that there are certain proteins that are expressed, such as calreticulin, release of ATP, turn on of HMGB1. So I will review some of the data that shows that our platform is really good at doing this, and that we are now moving forward to try to treat human beings with terrible cancers by using this platform. Next, please. So for about 25 years, we as scientists have been trying to design viruses by trying to figure out what genes to change and what genes to delete. And our last generations of viruses were good, but not great. So in about 2015, a number of us decided to redesign viruses now that we've learned a lot about how viruses work. So in our group, what we did was this. We took a bunch of vaccine viruses that normally have been used to go vaccinate human beings to protect them against natural diseases. And we put a bunch of these viruses all together into the same cells. Viruses live to trade genetic material, and therefore, after a few days, within these petri dishes were all kinds of new viruses that never existed before, with pieces of DNA from multiples of these original vaccine viruses. We then chose 200 of them, grew them up to be a large cohort of viruses to test. Then we put it through something called high throughput screening, meaning that we actually put them into a machine where we actually look to see how well these viruses killed an entire group of 60 different cancers that are used by the NCI, the National Cancer Institute of America, to screen for agents that could kill cancer. And we actually said we only cared about the viruses that only killed all of them, okay? There was a whole generation of virus design where we were trying to design viruses that only kill brain cancer or colon cancer. But in this generation, in my lab, we decided that we were going to go only care about those that killed everything in the NCI 60. And then we took the ones that we found that did that and found which ones were safe in animals. We injected them into animals to make sure that even though they killed cancer really, really well, that they didn't really harm animals in a reasonable dose. And then we added all kinds of other genes and proteins to it to make the viruses even more robust. And this was a whole group of viruses called Hope oncolytic viruses. And the best one we found was a virus called CF33. Next, please. When we gave this virus to animals, animals that had various kinds of cancer, it could be pancreatic cancer in the upper left, colorectal cancer in the upper right, colon cancer in the bottom left, or lung cancer in the bottom right. Normally, the tumors would grow in the flank and get bigger and bigger at those are the top lines in each of these graphs. But as soon as we injected these tumors with a very small dose of virus, what we saw was that many of the tumors regressed and some of the tumors actually disappeared completely. And it was quite remarkable. And we were able to do that sometimes with as little as 1,000 particles of virus, which is really a teeny tiny dose. To put that in perspective, in the last generation of viruses, we generally had to go up to over 1 million viruses or 100 million viruses in order to go and see similar effects. We're now at a dose level of about 1,000. And then on the right there, it actually shows us the agent that we're going to be talking about today, which is HOV3, which is CHECKvacc. They design a virus that we have taken into human trials. And again, as little as 10 to the third, 1,000 viruses, that's the orange curve on the right, retarded growth of tumors in animals in a model of triple negative breast cancer. Triple negative breast cancer is probably the worst breast cancer that's out there. And we have very few chemotherapies that work, and patients with advanced disease generally all die from this disease. Next, please. We were also able to turn on the local immune environment. What do we mean by that? There is a kind of protein that turns on locally in immune cells and in cancer cells called checkpoint proteins that actually are hallmarks of how immunogenic a local area or immunosuppressive a local area is. And by injecting our virus, we actually can turn the areas much more with much higher expression of checkpoints such as PD-1 and PD-L1 that is shown in the upper right, with the brown in the tissue being much higher. We injected virus in the local tumor area. On the bottom right, we see that by injecting our virus into tumors, we can actually attract the kind of lymphocyte called CD8 positive lymphocytes. These are the immune cells in our blood that is the best at targeting and killing cancer, and we actually attract them to the sites of cancer by injecting virus there. This is animal data, and you can see along the bottom left that we could truly turn on the local immune environment, attract a lot of cells that could come and kill cancer, and these were very, very encouraging data in animals that led us to our human trial. Next, please. We also put into our virus a gene called HNIS. That stands for human sodium iodine symporter. This is a normal human gene that's in all of our thyroid gland. Our thyroid gland uses this gene to bring iodine into our thyroids so that we can make thyroid hormone. But any cell that expresses HNIS, we can actually find it by just giving to an animal iodine and radioactive iodine and scanning for the expression of HNIS. So if you look at the scans on the right side, that those are mice with tumors in the flank. We also show the thyroid, the stomach, and the bladder because those organs tend to have some natural HNIS expression, and that's why iodine concentrates there. But if you look at the upper left-hand animal there next to the 1E05 label, that arrow is pointing to a flank tumor, a teeny tiny tumor that's about a 1/4 inch in size or about a 1/2 centimeter that you can actually see by iodine imaging using PET scan that a virus has gone there, and therefore it expresses HNIS, and we can find where virus has gone just by doing imaging, non-invasive imaging of animals. The reason we put this gene in is so that we can track our viruses and learn a lot more during trials in terms of where virus goes. Does it correlate to where cancer shrinks? And does it correlate to where immune cells go? Next, please. This is an experiment where we gave our virus CF33HNIS, and then we also gave to the animal an immune therapy called anti-PD-L1. And by combining these two, we actually generate tremendous anti-cancer effects. So if you look at along the B line on the left-hand side, all those are single animals with tumors growing in the flank, and those tumors were treated with PBS, which stands for saline, so it does not include virus, and the tumors just grow and grow and grow. In the orange lines, you can see that we gave anti-PD-L1, and some of the tumors are slowed down. And in the green line, we gave a teeny tiny dose of our CF33 virus, and 2 of the 8 animals are cured, and many of the other ones are slowed down in terms of growth. But when we gave the combination, those are the purple lines on the right, 4 out of the 8 animals are cured of their disease. And in panel E down below actually shows us that those animals that are with their purple curves actually lived a lot longer. Those are survival curves. Half of those animals were cured of their disease and continue to live on. And therefore, quite encouraging data that virus not only kills cancer, but stimulates the immune system to be much more sensitive to checkpoint inhibitors and to be able to be used in combined therapy for a tremendous effect. And that's where we're going in future clinical trials. Next, please. They were also really safe. These are studies looking at toxicity, both by injecting the animals in the brain as well as injecting animals in the flank and in the bloodstream. And natural vaccinia viruses, as little as 100 particles into the brain would be lethal. And our virus, as much as 100% of the animals that got 100 particles survived and did well and did not lose weight. So we were quite encouraged that not only did we find a virus that was really good at killing cancer, but somehow this virus has lost natural ability to harm animals and to be much, much safer in mammals. Next, please. So this is our agent CHECKvacc, and I show a schematic of the genes that are modified in it. In the upper left, a gene called thymidine kinase has been disabled by our insertion of our HNIS gene. We did that both to have the HNIS function so we could track the virus, also to make the virus much safer. We disabled the thymidine kinase gene, which is a very important gene for viral replication. We also put into this virus a checkpoint inhibitor called anti-PD-L1 that's shown on the left-hand side there. And locally, where we inject the virus or where the virus lands, there's actually a tremendous expression of such checkpoints, and that's orange lines there. That's a protein gel showing the expression of anti-PD-L1 antibodies. And on the right again is really that we could track the virus. But down there on the bottom right, not only can we track virus, but when we combine our virus by 7 days later giving a small dose of radioactive iodine that is lethal to tumor, we can also greatly enhance the effects. And that's what those curves show down there. By giving virus and by giving a dose of iodine 131, which is a radiotherapeutic dose of iodine, we can actually cure most of the animals. And that's the line with the squares there. Next, please. So that's the science behind what we're planning on doing. Again, we have a virus that as little as 10 to the third, 1,000 particles can cure animals of tumors. And we did not see toxicity all the way up to 10 to the ninth. So 6 logs of what we call therapeutic index, or a window in which we can actually operate and and expect to be safe. We have added proteins that allow us to track the virus, and proteins that allow us to have the virus more effective in stimulating the immune system. And we are now moving forward to human clinical trials. And I'm going to turn this over to Dr. Yuan, who's the principal investigator of the trial, to explain the current trial that is ongoing. Yuan?
Okay, thank you, Dr. Fong. So thank you, everybody, for this great opportunity to discuss this very exciting study. So this is triple-negative breast cancer is notoriously for very resistant to chemotherapy, and even despite the recent FDA approval of immunotherapy, we still facing a very challenging disease with overall survival 8 to 13 months, as shown on the slides here. And only a fraction of patients would truly benefit from immunotherapy. They needed to have PD-L1 expression, for example, to qualify for this drug. So the single agent activity of atezolizumab, it just showcase how limited immune checkpoint inhibitor by itself. In the first line setting, response rate about 25%, and it quickly goes down to 5 to 6% in the late line setting. So that really speaks for the unmet need for this disease, and often we're facing young women with detrimental prognosis. So our trial is designed to testing the safety and efficacy of the oncolytic virus being given intratumorally direct injection in patients with metastatic triple-negative breast cancer. And currently we have already received one patient being dosed, and so far we needed to looking forward to more data, and hopefully in the near future we'll be able to report back to you regarding the efficacy. Okay, I guess I'll pause here.
Great. So Matt, I think we can open it up for questions.
Great, thanks very much to both Professor Fong and Dr. Yuan. As Leslie's alluded to, we'll move on to the Q&A. Once again, for anyone viewing today, if you'd like to submit a live question, please do so using the Q&A panel within Zoom, and we'll get through as many as we can. So the first question is for Dr. Yuan. Can you elaborate on exactly what triple-negative breast cancer is and why it's different?
Sure. So the so-called triple-negative breast cancer refers to lack of estrogen receptor, lack of progesterone receptor, and then lack of HER2, which stands for human epidermal growth factor receptor. So when we talk about the breast cancer, we have 3 different subtypes. This so-called triple-negative breast cancer accounts for about 15 to 20% of all comers, carries a relatively challenging prognosis because of lack of those receptors, indicating we cannot treat them with hormonal therapy or HER2 target therapy.
Thank you. Next question is also for you, Dr. Yuan. What is the current treatment experience of a triple-negative breast cancer patient in terms of what they go through in the treatment paradigm?
Excellent question, and quite broad question. So this is a broad question, so probably I could attack that from an early setting, early stage newly diagnosed. And with the most recent FDA approval of pembrolizumab in 2021, now we have any comers with 2 centimeter size and above, or anybody with newly diagnosed lymph node positive triple-negative breast cancer can receive a combination of chemotherapy made up of carboplatin, Taxol, adriamycin, cyclophosphamide, and on top of that receive 1 year worth of pembrolizumab. That's sort of a standard care. Now in metastatic setting, it gets more complicated. So we normally kind of build a treatment algorithm based on biomarkers. As we mentioned earlier, PD-L1 is one of the important biomarkers to look to identify patients who may potentially benefit from immune therapy. But if PD-L1 is negative, then we only have the standard care chemotherapy to give to the patient. And normally we would pick the agent patient has not received in the past 12 months. And then second-line setting, we do have a novel drug antibody conjugates, such as Trodelvy. And then there's a fraction of patients with triple-negative breast cancer has BRCA mutation. They can receive PARP inhibitor, but most of the patients would benefit from clinical trials such as this one.
Thanks for that detail. The next question is, again for you, Dr. Yuan. How is CHECKvacc different and what hope does it provide to patients from your perspective?
This is a really novel mechanism of action. That's why we're so excited. Now, in the solid tumor world, we already have one oncolytic virus being FDA approved to treat melanoma, and this is the first in class to treat triple-negative breast cancer. We're very excited to see, from Dr. Fang's explanation, you learned that this is a unique virus, not only killing the tumor by itself, also expresses PD-L1. So we're hoping with our current trial demonstrating the safety in the future, we may be able to build a combination therapy with immune checkpoint inhibitors, but we will need to go through the first step. So that's why I, as a treating physician, I'm extremely excited to see this go into clinic.
Great, thank you. For Professor Fong, a short question, why add a transgene?
So the two transgenes that we put in is number one, putting in the HNIS transgene allows us to track the virus. In most drug development, when you inject the drug particles into a human being, it's kind of a black box. Now, not being able to figure out where it goes is why oftentimes we need so many patients and so long to go and do the trials. We're hoping that if we can actually track the virus and know where it goes, knows that it goes to tumors and not to normal organs, and to correlate with where tumors shrink, that we'll learn much faster and hopefully move the trials along much faster. So that's why the HNIS gene. The second gene was inserted because, again, we are trying to combine this virus with a kind of a therapy called checkpoint inhibitor therapy. And by having the virus make its own checkpoint inhibitor, we're hoping that both things will arrive at the tumor at the same time and actually be a therapeutic advantage. So these two genes were actually added purely because we believe that it will help the trials and help the therapeutic effects.
Thank you. Another one for you, Professor Fong, is why is it a good thing that injecting CF33 causes the tumour to upregulate PD-L1? Isn't PD-L1 used by the tumour to evade attack by CD8 plus killer T cells, and the reason why CHECKvacc is armed with an anti-PD-L1?
So yes and no. If we did not plan on treating the patients any further with checkpoint inhibitors, you're absolutely right. Turning on checkpoints could actually be detrimental. But what's been seen in therapies with checkpoint inhibitors is that unless you have upregulation of the checkpoints, meaning PD-1, PD-L1, CTLA-4, CISH, and many others, that unless they are activated in the local site, checkpoint inhibition does not work, okay? And so by even upregulating the checkpoints a little bit, we tremendously increase the efficacy of checkpoint inhibitors. In fact, in one of the melanoma trials, just upregulating PD-L1 by 1% change entirely the response to checkpoint inhibitors. So we are doing it because there is very good evidence that when viruses turn a tumor microenvironment from cold, what we say called cold, which is non-immunoresponsive, to hot, which is immunoresponsive, one of the markers of that is actually changes in local checkpoint markers. And we actually think in our therapy, turning on these checkpoint markers is actually a good thing, and plan to go combine in those patients checkpoint inhibition.
Right, and a follow-up from the same person was, is it true that by adding a large transgene, the CF33 virus virulence is somewhat reduced?
So by adding, anytime you add transgenes, you change the character of a virus, okay? Our virus, CF33, original virus was extraordinarily oncolytic, meaning that it was really good at infecting and killing cancer. And again, at as little as 10 to the third, it was able to kill and eradicate cancers in animals. And by adding these transgenes, though, we're hoping to get other therapeutic effects. And so on the one hand, yes, every time we add a transgene, we worry that we may attenuate the virus. But this virus is so potent to start with that we think that by adding 1 or 2 transgenes, we're still okay. Whereas in the last generation of viruses that were designed in the 1990s, the viruses were so attenuated to start with that adding anything sort of just made the viruses not work very well. So we're hoping that we will still see very great anti-cancer effects. But that's also why we are in our second trial going to go take a virus that only has 1 transgene added instead of 2. And we're going to actually know how much difference there will be between the 2 viruses.
Thank you. The next question is, do you think that this treatment will work equally well with colorectal cancer? And if so, what type of colorectal cancer do you suspect it will work best on?
So we actually have data from animals that our viruses kill colorectal cancer very, very well and turn on the local checkpoints in order to make it sensitive to checkpoint inhibition. We have all that data. In fact, we have that data in animals that it's not just the tumors that have a lot of mutations in it, but even tumors that don't have huge number of mutations, we can be effective in. Animal and human data are different, and therefore we are going to go seek human data to verify this and to try to move it forward for colon cancer. Our first trial here is in triple-negative breast cancer, but we actually have planned and are moving forward through the regulatory processes, a trial for many solid tumors as our second trial. And that will include in it colorectal cancer patients. So I think I'm hoping that by next year, we'll actually have data in that area. As you may or may not know, one of the things I treat the most of in my clinical practice is colorectal cancer. It is a cancer of great need, and it's a cancer of great interest to me.
Thanks again, Professor Fong. And we've just got time for one more question, which is, how long does the CHECKvacc virus persist after the solid tumour injection? And does it replicate and expand to more virus copies, or will repeat injections ultimately be required?
So in animals, when we have many animal models where we put a flank tumor on one side and a flank tumor on the other side, and we will only inject one side and not the other. And in about a week, we will actually find virus in the opposite tumor that was not injected. So we know for a fact in animals that the virus will replicate, will go into the bloodstream, not cause much toxicity, and travel to the other tumor site, replicate, and actually kill cancer and eradicate tumor. So we actually know in animals that happens. Whether that happens in man or not, we're about to find out. Okay, and that's because, again, being able to track these viruses now with HNIS, we're going to be able to tell where it goes after its initial injection. In our second trial, one of the arms is going to be intravenous injection. We're going to inject the virus intravenously in patients who have multiple deposits, and then try to figure out which deposit did it go to, and did those deposits that the tumor got to, a virus got to, shrink. So we are hoping to have human data in the next year or two that will actually answer all these questions. But most of all, we're hoping that these tumors will be smaller and the patients will benefit and the patients will get better. And obviously, that's a goal for all of us who are in cancer therapy, is to make a difference in human cancer outcome.
Great, thanks again. As I mentioned, that's the final question for today. I want to give a big thanks to Dr. Yuan and Professor Fong for taking time out of their busy schedules to be present for us today, as well as thanking everyone who joined for our inaugural Science Series event. We had a lot of interest and we really appreciate it. And we'll look forward to bringing you further instalments of the series in due course. But But that's all for today, and you can now disconnect.
Thank you.
Thank
you,
you.
Matt.
Thank you.